Hello students, welcome to uttyoron. Now we were continuing with the lecture on diversity in living organisms, diversity in living world, that is the name of the chapter. Today we will be discussing about the first kingdom, that is kingdom Monera. In the previous video we have discussed about the five kingdom classification. Gradually we will be discussing about the kingdoms individually. Alright, so let us start with the first kingdom, that is kingdom Monera. So as said previously, kingdom Monera consists of prokaryotes. Prokaryotes only. Alright. So among the five kingdoms, one kingdom consists of the prokaryotes, rest four kingdoms from Protista onwards till Animalia consists of eukaryotic organisms. It forms the decomposer or biomass of the ecosystem. So bacteria, it forms the majority of the decomposers found in the ecosystem. Bacteria is a cosmopolitan organism also. That means it is present everywhere. It is present in extremely harsh environmental conditions to more suitable one. I mean, any place you can imagine the presence of bacteria. It is omnipresent, sort of. So, uh, bacteria is that, and nuclear materials are freely suspended in the cytosol. Now, if I come to the cell structure of the bacteria, which I'll be discussing in details a bit later. But since this point is here, let us talk about it. Nuclear material is not enclosed. It is lying freely in the cytoplasm because it is a prokaryotic cell. And the primary feature of prokaryotic cell is the cell organelles are naked. There is no specific membrane-bound cell organelles performing the respective functions, the one that we find in eukaryotes. But in case of, since bacteria is a prokaryote or the group of monerans or the kingdom Monera is a group consisting of prokaryotic organism. So for the members of Monera, nuclear material is freely suspended in the cytosol. The cell wall is made up of peptidoglycan, teichoic acid, and Brown's lipoprotein. So these are the constituents we find in the cell wall of the members of Monera. And they have 7ts ribosome unlike eukaryotes. So remember, for eukaryotes, we have 8ts ribosomes. Whereas for the prokaryotes, we have the 7ts ribosome. Now let us talk a bit about the structure of the cell of bacteria. We will be there are other groups also that we will name in the end of the lecture, but the the prominent one that you need to know, the prominent member of the group that you need to know is bacteria. Okay, so the cell structure of bacteria you can see over here. There is a capsule present on the exterior, the outside. It may be present, may not be present. Then we have the cell wall. Now this is not cellulosic. It is made up of, it's a non-cellulosic cell wall made up of peptidoglycan. Next, we have the inclusion bodies and ribosomes. I said that they are made up of 70S ribosome, and there is a flagella present. There is cytoplasm inside the cell, and cell membrane is normal. There is nucleoid. Now, what is nucleoid, students? Nucleoid is the genetic material which is a circular DNA present inside the cell. It is the genetic material that is lying naked in the cytoplasm. It is not enclosed by any membrane. It has another name also, that is incipient nucleus. Okay. And lastly, we have the mesosome. Mesosome is an invagination in the cell membrane of the bacteria, which is responsible for the functioning, for the respiratory functions. You can relate mesosome with mitochondria. Like in our cells, in the eukaryotic cells, mitochondria is the seat of respiration. Here, mesosome is the seat of respiration. Next, let us talk a bit about the cell wall structure of the bacteria. Now, I have told you that it is not made up of cellulose. So, let's see what it is made up of. It is made up of, uh, it is having a polysaccharide, or you can say a sugar backbone, and it is connected. These sugar units are connected by amino acids, peptides. Okay, so look over here. So this peptidoglycan is made up of N-acetyl glucosamine and N-acetyl uramic acid. These two are the sugars that are present in the cell wall, the structure of the cell wall. They are interconnected, like there are chains dropping from the sugars, which consists of these amino acids. L-alanine, L-glutamine, L-lysine, D-glutamine, L-lysine, and D-alanine. These are the four amino acids found. And these amino acid chains are in turn interlinked by a pentapeptide cross-linking. Okay, this is not tetrapeptide, this will be pentapeptide. 1, 2, 3, 4, 5. 5 peptides are there, so it's pentapeptide cross-linking. All right, so this is, it's like, if I have to simplify, suppose these are the sugar units, there is amino acid chains dropping from it, and these are again interconnected by the next layer of sugars. So it's an interconnected network like structure, that is the cell wall of bacteria. It's not a very, it's not a constant structure. It's not like, you cannot imagine it like a brick wall. It's not like that. It's an interconnected structure made up of different compounds. So, what are the constituents of peptidoglycan? You would say it is the N-acetyl glucosamine and N-acetyl uramic acid. All right. Next, a little bit more about the members of Monera. They exhibit cell division by mitosis. So since they are exhibiting mitosis, so whatever steps we find, the steps of cell division we find in the mitosis, those are not visible in case of bacteria. Reproduction is primarily asexual. So bacteria mainly reproduces by fission or by amitosis. But whenever it is to whenever it is to undergo genetic variation, what happens? It performs conjugation, transformation, and transduction. Conjugation, I've given a bit of hint in the previous one of the previous videos where I've talked about sexual reproduction. So conjugation is not true sexual reproduction. Check the next point, students. They are saying that there is no true sexual reproduction taking place, but genetic recombination takes place. And how is the genetic recombination taking place? Obviously by means of these methods: conjugation, transformation, and transduction. So principal hereditary material is the nucleoid. I've shown you in the diagram what is the nucleoid, right? This is the nucleoid. This one. This is the nucleoid. So that is the principal hereditary material. The DNA, the circular DNA, is placed naked in the cytoplasm. Next, we have the accessory DNA fragments, which may be present or may not be present. Alright, so there are some other accessory fragments of DNA present inside the bacterial cell. What are those? Plasmids and episomes. Now, I would like to point out one difference between them. So much details is not required right now. Plasmid and episome, there is one difference. One main difference is that plasmids do not get integrated into the nucleoid. Nucleoid is the main genetic matter, main DNA. So plasmids, suppose a fragment, consider it as a fragment. Suppose this is the main DNA fragment and plasmid is another small accessory fragment lying somewhere inside the bacterial cell. Now, if this gets integrated in it, it will be called episome. And if it does not, then it is just plasmid. So what can we say? All episomes are plasmids. All episomes are plasmids. But all plasmids are not episomes. Alright? Reverse is not true. All episomes can be plasmid. They are all accessory DNA fragments. But a plasmid, when it gets integrated into the main genetic material, it will be called episome. Okay. So this was all about the internal structure of bacterial cell. Now let us talk about the kingdom Moneran subgroups. What are the different subgroups, the division that we have in kingdom Monera? So kingdom Monera can be divided into two major groups, Archaebacteria and Eubacteria. Alright, these are the two major groups of Monerans found. Now Archaea means ancient bacteria. You can remember it like this, A with A, okay? And Eubacteria is the true bacteria. You know the meaning of eukaryote, right? Why is it eukaryote? Eu means true, karyon means cell. So eubacteria means true bacteria, like that only. So archaebacteria or the ancient bacteria, and you can relate and remember it like this also. See, ancient bacteria means from the name we are able to understand that they have been present from the very beginning when the earth was young, just after formation, when the life forms came into existence. Ancient bacteria probably came in the, I mean, a lot earlier in the time zone, in the time period if we take ancient bacteria came a lot earlier. Now, when the ancient bacteria came into the existence, the conditions of the earth was extremely harsh, isn't it? So they have survived those harshness and they are surviving till now. All right. So remember it like this. The groups of bacteria which are there surviving the harsh conditions, the extreme conditions belong to the group Archaea. Let's take the examples. We have methanogens, we have halophiles, we have thermoacidophiles. So what is methanogens? Methanogens are the ones which are decomposing and producing, which are able to survive in extreme anoxygenic conditions. Okay, they don't need oxygen at all. They are obligate anaerobes. These are, I don't have space to write. Please note it down. They are obligate anaerobes. Next, we have halophiles. Halophile means salt loving. Salt loving. All right. So salt loving means they will be existing in the conditions of extreme salinity. Example we have over here. Next, there is thermoacidophiles. Now, I was talking about the thermoacidophiles in the brief introductory videos that we have. So, thermoacidophiles. Thermo means related to temperature, and acidophile means acid loving. So, they exist at a pH as low as 2. and they are able to exist at temperatures up to 110 degrees Celsius. So you can see these three groups, you can already get from the names only. They are able to survive in the extremely harsh conditions. So archaea or archaebacteria represent the groups of the bacteria which survive in extreme harsh conditions. Ancient bacteria, they have survived in the ancient earth, which was not very comfortable as it is today, although due to our irresponsible activities, we are gradually making it a bit more uncomfortable than it should be. But uh, these discussions apart, ancient bacteria, they have lived on the ancient earth, which was not very comfortable as it is now. So they are capable of surviving the extreme conditions and they do exist in extreme habitats. Next, we have the eubacteria or the true bacteria, under which we have the bacteria, we have mycoplasma, we have cyanobacteria, we have actinomycetes, we have rickettsia, and spirochetes. Please take down the examples that are mentioned over here. And among this, what mentioning is mycoplasma? See, whenever you're suffering from any sort of bacterial diseases, you're usually prescribed antibiotics. What does it do? It messes up with the composition of the bacterial cell wall and kills it. But mycoplasma is a group of bacteria which does not have cell wall. So how will you kill it with an antibiotic, right? So mycoplasma is worth mentioning among this group of eubacteria. Cyanobacteria is probably the earliest colonizer of the earth. Earliest colonizer. They are able to photosynthesize and they also sometimes have a special cell, special structure called heterocyst, which enable them to fix nitrogen. The example which we have over here is nostoc. Nostoc, Annabina, you have learned these examples from your junior classes. They are all nitrogen fixing bacteria also. So that is all about the kingdom Monera. Now let us take a few questions to test your understanding. Which of the following belongs to kingdom Monera? So here some of the name of the genuses are given. Some genus has been mentioned here in the different options. You have to choose which one belongs to the kingdom Monera. So first one is amoeba. No, amoeba is not a prokaryote first of all. So even if you don't remember which kingdom amoeba belongs, it is clearly not a prokaryote and hence it is not part of Monera. So amoeba cancelled. Next we have Gelidium. Gelidium is an algae. So this is again, I think it is a red algae more specifically. So Gelidium is again not part of kingdom Monera. Spirogyra is also an algae, not a part of Monera. Escherichia coli. I mean, here only Escherichia is given, but you know the name from E. coli, commonly called E. coli. You remember this organism very well, and that is a bacteria causing a lot of diseases. And this is a bacteria. So the last option is Escherichia. We know the example Escherichia coli or E. coli, right? And that is a bacteria. So this genus Escherichia belongs to the kingdom Monera. So our correct answer is option D. Next we have halophiles are bacteria. So what is the definition of halophiles? If you know the definition of halophile, you will be able to answer this question very easily. Halophiles, what did I say? They are the salt-loving bacteria. So let's check the options. Surviving in low pH, no. Halophiles have nothing to do with pH. Low temperatures, nothing to do with halophile. High temperature has nothing to do with halophile. Extreme saline conditions, yes. They love salt, and hence they are capable of surviving in the extreme saline conditions. So that's all for today, students. Hope you've understood the concepts well. Please stay connected with Uttyoron on different social media handles and please revise your concepts. Thank you.